Okay. Good afternoon, everybody. My name is Ilana. I'm the director of the MJC MESA program. I have the great honor to present today's science colloquium. So today's presenters are four groups of MESA students. That's a program here at MJC who have been working all semester on research projects related to the circular bioeconomy. Well, many of today's presenters will talk about what this means. Basically, all these projects have to do with how we can utilize biological materials and waste products to create and maintain economic opportunities and improve sustainability. Our region is especially well situated to be a leader in this area, and we hope that these projects have inspired our students to continue this work in their future endeavors. This work would not have been possible without the support of Beam Circular. I've been told they're on their way, they're a key player in this new industry throughout our region. I'd like to especially thank Eric from Beam, who worked with me to make sure we could make this project happen. I'd also like to thank our faculty mentors, who I think are almost all here. Noah Hughes, Aaron Thompson, you can wave. Thank you. Georgette Araha, who you already just saw, and Rick Estrada. Also, our research librarian, Kathleen Innes. I know how thinly you guys are all spread. Thank you so much for believing in this project and for all the time that you have dedicated to your student groups. So after each group presentation, there'll be 1 to 2 minutes for questions. So we'll take a couple of questions between the groups as the other group sets up. However, if you have more questions that we're not able to get to between the sessions, please feel free to stick around after the talks. We have these posters up, and all the students will be standing by their posters, and we'll be able to talk about what their projects were and answer all those questions after the symposium. So without further ado, I would like to introduce our first group, Mariana, Maria, and Jessica, if you guys would like to come up. You just kind of pass this microphone. And so it goes onto the... Yes.
Good afternoon.
Oh.
They on? Really? Oh, good afternoon. Can everyone hear me?
Yes.
Yes? Okay. Alrighty. My name is Mariana. These are my teammates, Jessica and Maria, and today we will be presenting the bioremediation of industrial wastewater using agricultural waste. Let's get started. When we think of our water footprint, we think about common tasks such as watering our grass, washing our cars, washing our hands. However, the reality of our water footprint, which is the volume of water consumed to create goods and services specific to a particular person or business, extends far beyond these daily interactions. In the United States, we produce an average 444.3 billion cubic meters of industrial wastewater a year. 47% of that wastewater is industrial, which means we have one of the highest water footprints worldwide, which we need to address in order to maintain ecological equilibrium with our environment. Throughout our next slides, we will explain how bioremediation can help address this issue. But first, Maria will introduce some key terms that we will be seeing throughout our presentation.
Thank you, Mariana, for that lovely introduction. Okay, so let's talk key terms. So let's start with industrial wastewater. So industrial wastewater is dirty water that comes from factories or industrial places after they use it for things like cleaning or making products. So this water can have harmful chemicals in it, so it needs to be cleaned before you can reuse it or introduce it back into the environment. And a way we can do this is through a process called bioremediation. So bioremediation is a way to clean up pollution using living things like bacteria, fungi, or plants. Now, these organisms break down harmful chemicals, turning them into safer substances. We can source the necessary bacteria, fungi, or plants right here in the Central Valley, where we have agriculture, which means we have agricultural waste. So instead of just throwing these away, they can be repurposed in cleaning wastewater, which we'll talk more about in some later slides. So now that I've introduced you to these key terms, let's talk about how this is all connected to the bioeconomy. So the bioeconomy is combining biology with economics and for sustainable practices. So we now know that here in the Central Valley, we have agriculture, which means we have agricultural waste, which we can use to bioremediate wastewater. Once that water is clean, we can use it to produce things like water for irrigation, electricity, and the leftover sludge can be used for fertilizer, which consumers will buy, industries will make money, and that income will be put back into the industries, making the bioeconomy cycle. Now, Jessica's going to talk more about how exactly we're able to use agriculture to clean up the wastewater.
As we were researching this topic, a common occurrence we came across was the use of microbes. The first way to use microbes is through enzymes, which are natural catalysts that speed up chemical reactions within a living organism. By using this, we can latch onto pollutants and start breaking them down into less harmful substances without it being consumed in the process. The second method was through absorption. So pollutants will be stuck to the microorganism and can no longer move freely. Once they are stuck to the surface of the microbe, they can either be broken down or safely removed. Some examples of absorbents include potato peels and corn cobs, all of which can be found in excess here in the Central Valley. Now for some methods in terms of technology. The first method is using biosensors, which helps detect living organisms and monitor the bioremediation process. The second method was through bioreactors, which helps regulate conditions and pH and oxygen levels. The third way is through modified organisms. So we can take something that might not be as efficient and genetically modify it so it can break down things more easily. Now we'll go over some case studies where these methods have been applied.
Thank you. Now we will look into some real-world applications of bioremediation. We will first look into the Pennsylvania Living Filter, which address waterway pollution that threaten local ecosystems and farmland. In 1962, scientists at Pennsylvania State developed the Living Filter, which used effluent from the university as artificial rain to irrigate the surrounding farmland. In this way, microorganisms would break down pollutants through absorption, and it would gain nutrients such as nitrogen and phosphorus in the process. Some of the benefits that were seen were an increase in soil fertility, overall reduced pathogens in wastewater, and an overall well-being in the surrounding environment. Now, after nearly 60 years of the Living Filter, it continues to remain fully operational in the premises of Pennsylvania State, and it continues inspiring sustainable water treatment worldwide. Now we will look into the Turkish white rot study, which is a wood-growing fungi that removes agrochemicals from wastewater. Scientists in Turkey saw a decline in fish populations as a result of excess sludge caused by the agrochemicals. This was prevented photosynthesis from occurring, which disrupted the overall ecological system. Is there a question? Oh, okay. Which disrupted the overall ecological system in the river. What was seen in this study was that white rot was able to decolorize 30 to 48% of wastewater. However, it was able to decolorize even more with the introduction of cotton stalks. Decolorization went from an average 37% to 71%, which demonstrated that diverse organisms can collaborate with one another to enhance pollutant removal in water ecosystems. Now I will briefly discuss the Indian River study, which, much similar to the previous study, assessed the effectiveness of two algal strains, vulgaris and turpin, in removing biodegradable and non-biodegradable material from wastewater. What this study found was that vulgaris was able to remove 70% of non-biodegradable material and 81% of biodegradable material. Turpin achieved an 89% reduction in biological oxygen demand and a 61% reduction in chemical oxygen demand, improving water quality by reducing organic and chemical pollutants from wastewater. Although the majority of these studies were conducted in a controlled environment, they were successful in accomplishing bioremediation, which by using agricultural waste as a resource. Similar approaches can be adapted to industrial wastewater treatment, offering a sustainable solution to this issue in our region. Now that we've looked into case studies, let us now talk about the challenges we face here in the Central Valley.
Okay, so a challenge that we face here in the Central Valley are the mixed pollutants. So as industries produce new contaminants like microplastics or pharmaceuticals in their wastewater, bioremediation must adapt to target these new pollutants specifically. Now, the mixed pollutants are hard to remove, and wastewater treatments often fail to remove them effectively. Another challenge that we face here in the Central Valley is the Mediterranean climate. So here on the bottom left, we can see a picture of California during the summer. And we can see how hot and dry we are during the summer. And I'm comparing it to Pennsylvania, who doesn't seem to have the same problem. So the dry climate and the periodic droughts that we face here limit the water resources and reduce nutrient availability. Agricultural waste can help compensate for limited water and nutrient availability that we face. So unlike Pennsylvania, we need to consider both drought and mixed pollutants. Now, let's talk about what could be done. So how Mariana was saying that in the Indian River study, it showed how vulgaris and turpin excelled at removing pollutants. This suggested that identifying or engineering specialized species may enhance pollutant breakdown. Now, in the Turkish white rot fungi study case, it showed how fungi and natural materials work together to remove pollutants. This shows the potential of hybrid techniques for greater efficiency. Now, another area we can look into is testing hybrid systems. So hybrid systems combine bioremediation with other technologies, such as filtration, which cleans water by removing pollutants through a physical barrier, or UV treatments, which use ultraviolet light to kill pollutants. Now, this way we can target multiple pollutants at the same time. California's investment in sustainable technologies creates an ideal testing ground for hybrid solutions. Now, over to Jessica, who's going to help us envision the future.
There are many benefits to bringing a circular bioeconomy to the Central Valley. The first one a lot of people like to think about is money. So what was once considered trash now has an added value and is turned into our capital. The second benefit is water security. As you heard before, the Central Valley struggles with droughts, and through the bioremediation of water and using reclaimed water, we can worry less during these disasters. The third benefit, which I like to think about the most, is our community. There would be collaborations with Beam Circular, Stanislaus 2030, and Berkeley Lab, as well as UC Merced, to bring higher quality jobs here to the Central Valley. Lastly, our environment would benefit a lot through sustainable practices by not letting our waste go to trash, but giving it a second use, which in turn reduces our carbon and water footprint. Now, I want you to take a look at a map of the Central Valley. And here is a second map of California with the underserved areas highlighted in red. If you notice, where we are in Modesto, we're highlighted in red. and it does not have to be this way. Also, if you notice, the Silicon Valley hardly has any red, probably because of their technological hub. Here we have agriculture, and we can bring biomanufacturing jobs here to the Central Valley, and people no longer have to drive all the way to the Silicon Valley for higher quality jobs. Thank you all for coming, and a special thank you to Elena Peachfine, Beam Circular, Georgia Araha, and Modesto Junior College, and we'll take questions now.
Thank you.
Thank you.
So where is the presentation? It doesn't mic, it just goes to there. So just make sure you're connecting your eyes. Where? Okay, so this is you guys. All right. Okay. Sorry, I'll take There you go. There you go. So I don't know how you guys are gonna, it has to be a little bit, there we go. That should work. Is it on the board? It's not on the board. That was my question. Okay. Very good. You need to display that. I think this is their device. Yeah, this is because theirs is on a PowerPoint.
Okay,
So
and so what's,
it's just not showing like the projector was.
oh, okay.
Need to display it.
Yeah, so right now it's under extend display, so we want to duplicate this.
Okay, perfect. Thank you so much. I was like, what was happening? Yeah, theirs is not.
There you go.
Doesn't do it on my-
Is that the way you want on those two screens?
Yes. Yeah. Okay. Thanks, Noah.
She's taking a picture. I want to wait for Elena. I guess I get their attention now.
So our next is Alexandra and Noah, and I will let them take the stage.
Hello, everyone. My name is Alexandra Richardson,
I'm Noah Vandenberg.
and we are part of MESA, a program here aimed at preparing and supporting STEM students. Thanks to our organizer, Elena's help, we were able to partner with Beam Circular this semester to bring attention to the need for circular bioeconomy practices here in the Central Valley. My partner and I focused on algae's role in this system, and specifically the potential that algal biofactories have. Earning the nickname green gold, algae is a precious and valuable renewable resource. But unlike gold, algae is renewable and can be used in a wide variety of applications. As one of the most promising bio-based materials, it can minimize waste and maximize value when used efficiently. With that being said, let's get into it.
All right, so to understand algae's role in the circular bioeconomy, it is important to understand why exactly we need it. The circular bioeconomy is a process desperately needed by California, particularly the Central Valley. This figure highlights the 40% of the state referred to as the Central Valley, expanding 400 miles in length with 18 different counties. Living in the Central Valley, I'm assuming we're all familiar with its agricultural influence, producing 34% of the nation's fruits and vegetables. Its fields, orchards, and of course, smelly dairies are vital to our communities. This is thanks to the rich soil, mild climate, and water availability in the state, all of which plants need to grow efficiently. The Central Valley holds 50% of California's water supply with 63,000 miles of water and 800 square miles of lakes and reservoirs. Combined with ideal soil and climate, it is central to the state's agricultural success.
However, this doesn't come without a price. Conventional farming, popular for its high yields and low costs, relies heavily on synthetic chemicals like fertilizers and pesticides. While efficient, this degrades the soil structure and pollutes surface and groundwater, impacting the environment and our health. Of the 63,000 miles of rivers and valleys mentioned just a slide ago, 1,500 of those are dominated by agricultural discharge. This refers to the combined use of chemicals and irrigation, producing a runoff of pesticides and heavy metals contaminating surface water, groundwater, and our drinking water. This is cause for concern, in addition to tillage and heavy machinery. Tillage disrupts the soil structure, causing erosion and unnecessary carbon release into the environment. These heavy machineries also rely primarily on fossil fuels for energy, also increasing the agricultural carbon footprint.
In addition to produce farms, the dairy sector is another cause for significant greenhouse emissions. The Central Valley's dairy sector, with over 1,000 dairies and 80% of California's dairy cows, produces 41.9 billion pounds of milk annually, making California a leader of dairy products. However, this success comes at the cost of significant greenhouse emissions. Methane, a potent greenhouse gas, produced through enteric fermentation in cow stomachs, primarily released through belching, with each cow generating up to 264 pounds of methane. California's 1.7 million cows produce about 448 million pounds yearly. So how can we reduce the negative impacts that this has on the circular bioeconomy? One potential solution is algae playing a key role. To understand the full potential of algae, it's important to understand what this model is. With some innovation, the circular bioeconomy shifts the traditional take, make, waste model into a circular one, where waste becomes a new resource for sustainable production and regeneration.
However, the circular bioeconomy cannot stand on its own and is made up of two components, the circular economy and the bioeconomy. The circular economy is a system that is regenerative by design and focuses on three key principles: utilizing waste, keeping products and materials in use, and reimagining waste systems for reuse. The bioeconomy, on the other hand, encompasses sectors using biomass to replace fossil resources, aiming for economic and environmental benefits. Therefore, the circular bioeconomy combines the what of the circular economy with the how of the bioeconomy, emphasizing renewable resources and waste reduction. By replacing fossil fuels, this system promotes sustainability, resource efficiency, and climate change mitigation, paving the way for a more economic framework.
But how does algae fit within this framework? Algae play a key role in the circular bioeconomy by providing sustainable solutions across various sectors, as you see on this slide, including biomass production, wastewater treatment, and CO2 sequestration. They can be cultured to produce a variety of byproducts that enable the cascading use of a single feedstock. Due to their rapid growth and adaptability, including in wastewater, their ability to capture and store carbon further supports the circular bioeconomy by reducing greenhouse gas emissions and further closing the carbon loop.
It is also important to understand exactly what is algae. Algae is one of Earth's oldest life forms and not classified as plants despite their ability to photosynthesize. They're actually protists lacking vascular systems such as roots, stems, and leaves. And they're primary producers. Algae produce oxygen and serve as a food source for various organisms. Currently, there are over 175,000 species of macroalgae discovered. These can be categorized into two sections, macro and microalgae. The macroalgae is what we commonly see as seaweeds, which includes red, green, and brown algae and can grow up to 200 feet in length. Microalgae, on the other hand, are microscopic eukaryotic organisms, including xanthophyta and euglenophyta. However, there's also another aspect we want to look at called cyanobacteria. While this is actually considered a bacteria, we use it as algae, and because of its ability to photosynthesize, that makes it really helpful for us in various sectors.
Photosynthesis is crucial to algae's success in the circular bioeconomy. It allows algae to act as a renewable energy source, capturing CO2, a key greenhouse gas. This process converts light energy into chemical energy, enabling algae to produce valuable bio-based products. Its ability to capture CO2 makes algae a sustainable resource for various applications.
This process can be optimized to increase yields of a specific compound through metabolic manipulation, which alters algae's metabolic pathways and boosts traits like oil production, growth, and stress resilience. Algae primarily use these three metabolic pathways shown. We have autotrophic, using light for energy, heterotrophic, using only organic carbon for energy, and then in the middle, we have the mixotrophic, the combination of the two, using both light and organic carbon. The mixotrophic pathway is one that we only see in algal biofactories, allowing algae to grow in diverse conditions and increase their metabolism, leading to a higher biomass production of a desired compound. This versatility makes algae a promising resource for industrial and environmental practices. To better understand how algae are cultivated and transformed into these valuable products, let's walk through the process. Algae biofactories use algae's natural ability to grow quickly and capture CO2 from the environment to produce biochemicals that are in variety. After a few days of photosynthesis, algae can already be harvested and concentrated into a slurry. This slurry is then pressed to extract valuable compounds like we see here with the screw press. These are then refined for use, and the remaining biomass is either repurposed back into secondary products or recycled back into the system for future cultivation. This is an essential aspect of the circular bioeconomy.
So this process utilizes two systems of algae growth. So on the left, we have open pond systems, which are used for large scale production. The top image shows circular ponds, which are large shallow tanks with a wide radius and continuous flow, chosen for like cost effectiveness. Below that is a raceway pond, a narrow channel with paddle wheels for mixing, offering greater efficiency. On the right, we have closed loop systems like photobioreactors, and these cylindrical systems optimize growth by regulating factors like light, temperature, and nutrients, ensuring higher productivity and better consistency than the open ponds on the left.
As previously mentioned, microalgae have many applications across many sectors. This starts with growing algae in wastewater for simulated nutrients while it simultaneously captures CO2 from the environment. This algae then produces a range of valuable byproducts that can be utilized in a cascading manner from high value to low value, another aspect of the circular bioeconomy. These applications include pharmaceuticals, food and feed, bioplastics, and biofuels.
So one of the first things that we came across in our research is the use of algae for biofuels. Sustainable biofuels are not only becoming a more popular choice, but can serve as a variety of essential alternatives in the energy sector. Most popular, algal biodiesel can be used to fuel heavy vehicles and machinery, helping to reduce greenhouse gas emissions. Biogasoline and bio-oil can also offer renewable substitutes for traditional gasoline and oil, while ethanol can be used for fuel blends like ethanol. E85, excuse me, further decreasing fossil fuel dependence in commercial vehicles. Algae lipids can also be used to produce bio-based kerosene for jet and rocket engines, providing cleaner, more sustainable option for aviation transportation, a major polluter of CO2. However, this sector is incredibly expensive and energy intensive to produce. So while most algal biofactories start here, they often transition into other sectors mentioned.
One of these other sectors include the use of algae in pharmaceuticals, a highly valuable algae substitute. Algae are rich in bioavailable vitamins, proteins, and minerals, including omega-3 fatty acids, which improve heart health, reduce inflammation, and improve brain function. These nutrients make algae a valuable addition to dietary supplements for overall health and well-being. Algae is also seen in a medical setting. We're seeing it being used in anticoagulants to reduce the risk of heart attack and stroke, as well as in agar hydrogels for controlled drug delivery and wound sterilizing agent. Certain algae compounds are also being studied for herpes 1 and 2 prevention. In one study, we looked at red macroalgae in vitro. When in contact with the virus, the algae's polysaccharides can bind to the HSV glycoproteins, preventing the HSV virus from attaching to the host cells. Essentially, algae act as decoys, mimicking the cell surface, which the HSV needs for initial attachment. This offers a natural alternative to combat herpes infections or enhance the efficiency of already existing drugs.
Algae also have diverse applications in food and feed. So seaweed is used in dishes, snacks, protein bars, and algae oils. And algae oil is a healthier, sustainable alternative to traditional cooking oils. Algae can also be processed into flours and thickeners for food and animal nutrition. Macroalgae provides plant-based protein and essential nutrients for pet food, livestock, fish feed. This is not only a cheaper alternative to other sources of protein, but it has been shown to reduce greenhouse gas emissions in the food and feed sector up to 80%.
In the bioplastic industry, macroalgae are becoming a sustainable alternative due to their fast growth and natural abundance, offering a biodegradable and eco-friendly solution to traditional plastics, which rely heavily on fossil fuels. Algae polymers like cellulose can be spun and blended with other natural fibers for a more sustainable fashion industry. Polysaccharides from algae are also being used in grocery stores, such as biodegradable films and coatings to increase the shelf life of food, or to replace unnecessary plastic packaging, such as grocery bags.
However, to integrate algae into a circular bioeconomy, several challenges must first be addressed. And these include space limitations, job opportunities, government funding, and also policy support. For both biofactory systems, growing algae requires a lot of space and resources to grow, and even more to scale up the production. Meaning it may compete with food sources and can actually harm biodiversity when converting available land into farms. Therefore, these factories typically operate on a smaller scale, creating less permanent jobs and opportunities within these production. This makes the algal biofactory industry a niche field. Scaling up these algae systems requires significant investment and high upfront costs, and a lack of public support hinder progress. These inconsistencies make it difficult for businesses to move forward with algae-based projects, making global adoption difficult.
However, the future of algae in the circular bioeconomy can still be promising. Driven by the need for sustainable solutions in food, energy, and environmental security, research and development in algae biofactories is increasing every single day and serves as a foundation for solving these limitations. Research is helping to advance our understanding and support for policies that recognize algae as a practical and renewable resource, therefore creating more opportunities for funding and permanent careers in the field. This ongoing research is crucial to replace non-renewable resources with algae and addressing issues like conventional farming, especially here in the Central Valley. With the right development, investment, and policy, algae can become a cornerstone of the circular bioeconomy, fostering sustainable growth and environmental benefits for years to come. We would like to thank Beam Circular and MESA for giving us the opportunity this semester to conduct this research. We are incredibly grateful for the opportunity and the skills we have gained along the way, which we're going to use throughout our college careers. We'd also like to thank our advisor, Aaron Thompson, for his support this semester, as well as providing some of the practical images shown of algae being used in wastewater treatment. Your support this semester has been invaluable in exploring algae as a sustainable resource within this framework and in our communities, as well as keeping us sane throughout the entire process. And finally, we would like to thank all of you guys for your time and attention today. Your interest and engagement is incredibly important and appreciated. But before we go, are there any questions?
I have to go on to the next.
Okay.
But you, again, you're welcome to stick around and check out the posters, ask questions after the presentation. Next group, we have Gerardo, Brianna, and so if you guys, this microphone, it just goes directly into the YouTube. Yeah, you can move around with it. It doesn't actually mic you, so make sure to project your voice. It's just connecting. Also,
okay. You want me to do the computer? Oh, yeah, I'll do. Oh, yeah, on the first one, yeah. Okay, put in the presentation. What? Can you put it? Yeah. Thank you. Good afternoon, everyone. Today, our group, conformed by Gerardo Diaz Castillo,
Luke Lorenko.
And Brandon Morales.
will present the quantitative assessment of biomass, utilization, and availability, especially the models and formulas that we can use in order to determine the calorific value of the biomass in the pyrolysis process. So first of all, what is biomass? Biomass refers to the agricultural waste that is used in order to create products. Some examples of biomass are olive husk and almond shells. The circular bioeconomy is important in order to use the biomass because it combines two terms together, which is the circular economy, which is reusing and reducing in a closed loop system, and the bioeconomy, which is creating products from biological resources. By combining both of them, we can create products from biological resources that are reused and reduced in a closed loop system. So pyrolysis is a key process in the circular bioeconomy due to that it converts the biomass by applying heat into other products, such as biocharge, which is used to create products instead of using fossil fuels, a bio oil that is used to create diesel, and syngas, which is used to create energy.
So in order for a biomass to go through pyrolysis, heat value and calorific value need to be determined. So in order to determine this, they usually. Thank you. So in order to determine this, they usually use a bomb calorimeter. Although in the study that we researched, they realized it's not ideal due to, so the biomass would be placed here in the middle and it would go under high pressure oxygen. And then if heat would be released into a water bath, but due to it being an exothermic reaction, they're determining combustion. So heat tends to get lost in this and can even lead to an incomplete reaction. Instead, what they used from the bomb calorimeter, they used approximate and ultimate analysis. So an ultimate analysis is the elemental composition of the biomass. So this is going to be carbon, hydrogen, oxygen, nitrogen, and sulfur. Key thing to note, carbon and hydrogen are very essential in combustion. So they mainly looked at carbon and hydrogen in most of the study that they were doing. And then for the approximate analysis, it's going to be the overall composition of the biomass. So that's going to be fixed carbon, volatile matter, ash content, and moisture. They're also very key in determining combustion of the biomass. So after conducting the approximate and ultimate analysis of the biomass, they use these analyses to compute empirical correlations. So an empirical correlation is going to be how does each variable relate to one another. So how does carbon relate to oxygen? How does carbon relate to nitrogen? And they tested about 400 different biomass feedstocks. So the study focused on forestry and agricultural. The one that we mainly looked at is going to be agricultural due to it. We're in the San Joaquin Valley and we produce majority of agricultural biomass. So after we use 400 different biomass feedstock, and then they split it up into 3 of them. So we already have the ultimate and proximate analysis, but then we have the combined proximate and ultimate analysis. So they also wanted to see how does carbon relate to volatile matter, how does carbon relate to moisture, and how do these relationships and variables, how is that important in determining HHV so we can use it for pyrolysis. So with the empirical correlations, they needed to see how do they relate to one another. So 45 different regression models were created out of this. This was the empirical correlations were turned into an equation, and which then turned into the 45 different regression models. But the question here is, how do we determine if the model is effective? How do we determine if it's even accurate? That's where they used 3 different statistical methods to determine error. Although they're all measuring the same thing, which is error, they're all very unique. The first one that they use is going to be mean absolute error. So this is going to be the sum of average of absolute error. So in this equation right here, n is going to signify the amount of samples used, and then the y and x is going to be the predicted and observed values. So it's basically taking the difference between the predicted and observed. This one is nice because it's very forgiving when it comes to error. So if we have a lot of outliers, it's not going to tend to penalize us as much. And it's also nice because we get the same unit of variables. So in the study, it's going to be kilojoules per gram because we're measuring energy yield. Okay, so then the next equation is going to be mean absolute percentage error. This is going to be the average of absolute percentage error. So this one is very similar to the last one, but it's going to be in a percentage form. Although this one is not as forgiving when it comes to error due to the fact that, it's giving it in percentage. So if we're underestimating or overestimating, it's not going to be too forgiving. We can't have negative percentage or over 100%. And so it's also taking relative of the predicted values to the observed, and it has high sensitivity to outliers, which is the error. And then the last equation used is going to be root mean square error. So it's going to be the square root of the mean difference between predicted and observed. So it's giving us a standard deviation of error. So
how far from the predicted are we going when determining HHV. In this case, the highest root mean square error of the model indicates a very good model. So, we're determining HHV very nicely. This one does have high sensitivity to error, but it's good because that's how we're able to pick out the outlier, see which one is not accurately determining HHV. So the study, they had 45 different regression models, but at the end, they picked equation number 10 to be the most accurate in determining HHV. The reason why is due to the R squared. So the R squared is going to be the correlation coefficient. So we had just mentioned that we did the empirical correlation, but this is saying how accurate is this relationship in determining HHV. Usually for R squared values, we want somewhere between 0.7 and above in determining a good model. This one had a 0.8, which is the highest. It also passed 3 of the error equations that we just covered right now. And if we're taking a look here at the regression model, we're seeing that there's very few outliers, and the red line right there in the middle is going to signify predicted, and the observed are very close to the predicted. So HHV is being accurately determined. So here for equation number 8, this is going to be not the worst model, but it wasn't very accurate in determining HHV due to the fact that we take a look at the R squared, very low, 0.2, meaning that whatever we're putting in the input is not accurately determining HHV. And we can also see that there's too many outliers out there, so HHV is not being accurately determined. So apart from linear partial regression, the study we also found was Monte Carlo based particle filter, which I will now give the stage to Luke.
Thank you. So to reiterate our objective with these models, right, we want to create some sort of mathematical model that'll tell us how much energy is in agricultural waste, right? And ideally you do that in a cost-effective and cheap way, and that's usually math. So Jerry, next slide, please. So the study we looked at utilizes a Monte Carlo-based particle filter to minimize these alpha values. And just to give you all a quick rundown of the study, using olive husks sourced from the Mediterranean, their contents of carbon, hydrogen, and nitrogen were analyzed. It's much similar to a process in how you see like protein or fiber content on a nutrient label at the grocery store. And from there, they actually determined the energy value in these olive husk samples. Right. And so the reason why we're using this equation in particular is because with chemical kinetics and things like combustion, there's a lot of non-linearity and it's a complex reaction. So as opposed to things that are normally assumed in the field like linearity or even in some cases normal distribution, we use a logarithmic model instead. But even though we have this equation with these concentrations that can be plugged in, we still need to determine these alpha values, right? So alpha 1 through 7, whereas some of them are coefficients, some of them are exponents. And that is where the Monte Carlo-based particle filter comes into play. And right, our whole goal is to create a generalizable model that we can use to predict energy in agricultural waste. So the way we're going to analyze the goodness of the fit in the model, the way that was done in the study was by minimizing this cost function here, where the L alpha is our predicted energy value and the LM is the actual energy value of the, in this case, olive husk biomass, but ideally we could use it for something like almonds, more local sourced. And again, our predicted values are going to be compared to our measured values. So on the left here, we have the pseudocode for the particle filter, and we're not assuming any sort of distribution whatsoever. We want this to be relatively random because it's the closest thing we can get to letting things happen in order to get a mathematical model. And to describe this, I'm going to use a process called Roomba math that filters out outliers. You guys will see on the next slide. So here's some diagrams that I made in Python in order to kind of illustrate this. This isn't the exact one that they use, but it's a random particle filter, not exactly a Monte Carlo particle filter. But as you can see here, we have these green X's. And think of those like walls, right? So this red star here is a Roomba, and you're sitting in a room and you're surrounded by walls, and you're trying to figure out where you are. So the Roomba is going to look around and trying to figure out where it is. And most of these dots you see are somewhat within the bounds of the X's. And the reason why I'm using a Roomba analogy is because the X's here are the greatest limits that this Roomba can be, or this data point that is the elemental composition being converted into energy that the data point can be in. So, right, and the reason why it's called a Monte Carlo based particle filter is because the Monte Carlo simulation is relatively random. It was named after a casino in Monaco. So we're randomly getting these points, and depending on how close they are to the red star right there, they get filtered, so the particle filter, over and over again until we get to a spread that's relatively close to the red star. So on the left here, we have the pseudocode for the resampling algorithm. And when Brianna, if you could go back one, but we need to still determine these outmost parameters, which are the minimum and maximum values that the elemental concentrations can contribute to the overall calorific value. And so by doing that, in order to determine these values in the study, half of the resources were used to compute the minimum value. So what's like the lowest, let's say, carbon can contribute to the overall energy. And the other point was used to search the space for potential ideal values. So based on the Monte Carlo process, that's how this was done. And the other half of samples were just assumed to be normally distributed when determining the parameters. And this was just to eliminate any sort of error. And so on the left here, we have the finalized equation where we're trying to predict our energy value in terms of carbon, hydrogen, and nitrogen concentrations. And we determined that in the study, it was determined by comparing it to the actual values. And ideally, where you'd go with future directions would be using AI or machine learning in order to hasten the process of parameterizing the function, because as you saw earlier, half of all resources were used trying to figure out where can these points be, and then finally the other half we were used to trying to figure out these coefficients. So my partner Jerry will now go into the benefits and challenges of these models.
Now, there's some challenges in creating these models, such as the data availability and accuracy, the model selection, and the methodologies. There's a lack of standardized methodologies, which can lead to varied results, and also accounting for parameters that we're not going to be able to know. But there's benefits in using them for determining the calorific value of the biomass, such as it helps predict the outcome on the pyrolysis process, such as how much biomass can be produced in the pyrolysis process. It enhances the optimization and management also of the biomass in the pyrolysis to create more products, and it aids in the decision-making of it. In conclusion, the quantitative assessment of biomass, the mathematical models that determine the calorific value of the biomass are very important because they transform waste into wealth, reverse environmental decline, and improve the eco-friendly economy. Thank you. Oh, one more thing. We want to thank MJC, MESA, and our mentor, Rick Estrada, and Elena. Thank you.
Do we have time for questions? Okay, yeah. Yeah, if you guys have questions, we're going to be here for the poster session.
Thank you.
Yeah.
This microphone doesn't mic, but it gets to the YouTube channel. So do hold it. So that people like you're good.
That's why we do that.
Second version. So I did that.
Okay, am I able to go to camera?
I think oh, yeah.
Don't have to send.
Is it really different?
Yeah.
It's really different.
Can we go back to my mom?
Yeah, I think so. I don't know. But
Yes, Dave, you can look at the presentation. You can see the notes.
No, but it's a different one. It's camera.
all right, I was just wondering if they could hear it on your laptop.
I don't want to connect it to that.
It's on their laptop. Yeah, they can hear it.
I mean, we could do the HDMI.
Yeah, sure.
We'll just have to switch the display here.
Okay.
It's a little different right now.
All right, so we want to do that in the HDMI
Can I use this one?
or
USB-C.
USB-C?
Yeah.
I think so, yeah, go ahead. And then we can switch that to the HDMI, so no input and then HDMI. Yeah, yeah. It should be that. Now that they did.
Well, that's HDMI.
Oh, okay. So you don't have, it's a USB-C, there wouldn't be that connection then. Oh, wait, USB-C.
It looks promising.
Almost there.
There's like, it has the menu at the top.
So we just need to drag it, right?
Yeah, I think so.
It's gonna go to all three projectors.
So we can do one that you can do not use, but I don't know if you do, like which one of these. would be good. I just know these are the ones that are,
Yeah.
if we don't want the center to show.
Let's go with the.
So it's one of these.
54 one. It's okay.
So I would assume it's,
I think we can go to. I don't need the notes anymore. We can just open this one and go off the
okay.
time.
We just need to drag it.
Okay.
Okay.
But my mouse.
You want to duplicate displays maybe?
Yeah. It would duplicate. It would be.
Settings.
Their settings. It's a Mac.
It's a Mac, yeah.
Oh, man. Let's go with the presentation. It's okay. Let's go with the PowerPoint, Professor. The other one. What's the difference?
For the Great Promise, it's different.
Yeah,
Oh, sorry. Yeah, I don't really know how to, I don't feel. It's okay. You don't have to just go with the Great Promise and that's it. Let's talk about anti-aging.
I think we have to use the other one, you guys. We'll skip it. It's taking too long and we only have 10 minutes left.
Room PC now? And then. And it's on this one, so I would just close this browser.
All right, sorry, guys.
No problem.
Why is that still?
We change stuff.
Better?
All right, so we're gonna finish up with our last presentation. Jackie and Idris, go ahead.
Great. Hello, everyone. Thank you for being here. My name is Jackie Quintero, and this is my partner, Grace Alzai. Our project was focusing on the local renewable businesses in the circular bioeconomy. So we have an overview, and we're going to go ahead and define what circular bioeconomy is, what the difference between a linear economy and a circular bioeconomy is, how the bioindustrial manufacturing works, and why the San Joaquin Valley is a great hub for a circular bioeconomy.
We're also going to go ahead and give an overview of the top 3 local renewable sources in the San Joaquin Valley and the products that come from them, and highlight an anaerobic digester project here at MJC, and the importance of the life cycle assessment to the circular bioeconomy, as well as the economic benefits for pursuing a circular bioeconomy. So what is a circular bioeconomy? A circular bioeconomy is an economy powered by nature that emphasizes on renewable natural capital while minimizing the waste and usage of fossil-based materials.
Hello, everyone. Again, my name is Idris. So basically, our goal is to change the linear economy to a circular economy. Basically, you have the source extraction, production, distribution, consumption, and waste. What you're going to do with waste now? You got to use this waste again and put it into the circular economy and make something else from it. So basically, there's a lot of benefits when it comes to circular bioeconomy. The first one is you have reduced the waste. Secondly, you make more money. And third, you have addressed the climate change. So there are 3 main benefits. There's a lot of benefits that we just want to focus on those 3 benefits. So basically, you take the waste, you convert it, and you make something else from it. So this is grape pomace. This is something else, and this is eggshells. So this is the plan. Here's the plan. We're going to go and take the waste, convert them, and make something else from them.
All right. So you may be asking yourself, why is that, right? So the Northern San Joaquin Valley consists of 3 counties, which consists of the Stanislaus, San Joaquin County, and as well as the Merced County. The Northern San Joaquin Valley was certified as a BDO zone, which stands for a Bioeconomy Development Opportunity Zone. So this was given by the Development Opportunity Zone Initiative, and it was also recognized as a AA rating, which has a strong capacity to meet its financial obligations. So this means if anything was to go down, it would be able to get back up. So this was given because it's amazing resources, manufacturing, which brings up the infrastructure, the agriculture, which brings us things such as almond, and the proximity to the Silicon Valley, which helps us with innovation, art, and technology.
You might ask yourself how much feedstock is available in San Joaquin Valley. So there are 4 million tons of waste by 2045, meaning there's a lot of waste, and we need to find a way to either get rid of them or make something else from them. It's a comprehensive list, but we want to focus on 3 biomass resources, which we are very, very excited about. The first one is the almond hulls, or I should say almond waste. So basically, we took the product, we have categorized them in energy, material, and food. So it's very fascinating that we can have bioethanol from almond hulls, and bioethanol, if we use the almond hulls and make bioethanol from them, we have reduced the fossil fuels, and then we have made it more sustainable. And also there's automotive headliners you can make just from almond hulls. Something that is really important is when you take the almond hulls and you mix them, can you change the slide, please? If you take the almond hull and mix it with cattle feed, you can reduce the methane emission by 95%. The second group talked about this CH4 and how cow are making a lot of CH4. But if you mix the almond hulls with it, you can reduce that CH4. So it's very, very important.
The next product we decided to focus on was grape pomace. So what is grape pomace, right? Grape pomace is the leftovers of the grape from grappa, from grape seed oil, which includes the skin and the stems. So some industrial materials that that can be made out of grape pomace are cosmetics, animal feed, soil amendment. Some food products can be grappa, seed oil, dietary fiber, and antioxidants. For energy production, we have biofuel and electricity. So for grape pomace, it is a great source of bioactive compounds that can be used in cosmetics, right? It concludes of things such as antioxidant, anti-aging, anti-inflammatory, anti-hyperpigmentation, photoprotective properties, wound healing properties, and organic acids. So this can be extracted by using nanotechnology. An example of this would be Foosh Beauty, which is a company that uses great promise to create biodegradable cosmetics. And then their packaging is also made out of wood.
Third on the list is dairy. Again, we have the same categories. We have the material, energy, and food. We can have ethanol from dairy, which is astonishing because ethanol if we use dairy and make ethanol from it, we don't have to import oil from other countries, and we don't have to invade another country to get oil. So basically, we have the dairy and we can just use it as an ethanol. This is the model by the MJC student. I like to call it the Cody's masterpiece, basically because it's an anaerobic digester. So this is basically the cattle manure, and they put it on a sealed tank, and they make a biogas from it. So basically, you can see the circular bioeconomy in action. So this is a great potential for materials like dairy. So see, we have the manure. It turns out to the bubbles, and then it will make pressure, and then you can have gas. So this is just a benchtop research model, but this is in large scale. So you can see that this has the potential to be used in a large scale too.
So for sustainability, we have three forces, which are environment, social, and economic, which make up the sustainability. Sustainability, people might have different opinions on sustainability. Sustainability is improving human well-being and ensuring social equity for present and future generations while safeguarding the planet by supporting ecosystems.
So basically, we can achieve sustainability by evaluating the product and see it through multiple impact categories. So basically, from the day we make it till the day it's over, so we need to see the life cycle of it and evaluate it, because if we have the product and we don't make the evaluation, it's useless. So life cycle assessment is really, really crucial. There are impact categories. So there are 13 impact categories. The first one is the global warming. Second one, the depletion of fossil fuel, ozone layer, acidification, eutrophication of water, and so on. So when you take a product, you need to evaluate it based on these impact categories. For example, going back to the anaerobic digester, when we use anaerobic digester, this will reduce the global warming first. Second, if we prevent it to go to the water, we have prevented from the eutrophication of the water. So it's a win-win situation again. So here is the, again, we want to focus on anaerobic digester, and you can see CO2 and CH4 is way, way lower than even pyrolysis, open burning, and landfill. So just by using the anaerobic digester, we have reduced the carbon emission and we have reduced methane emission. So again, you can see how circular economy, circular economy is really, really good, and it can have a great impact. Based on our research, we saw a great potential, a great need for standardization. We have talked with Eric, and also Gluramar, basically the beam circular, and we saw that the standardization is on their radar, and we encourage them to keep working on that one. And also we encourage every one of you guys to take active steps when it comes to biocircular economy. It's a promise, it's a great transition, and you guys need to be part of that. So basically, there's a lot of economic benefits. The goal is to make family sustainable jobs and keeping the youth in the valley.
Jobs can be like mechanical engineering, bioscience related job. But one of the top ones that we did get to see was the life cycle assessment specialist. This is really needed because we don't have a standardization. So having people who want to do life cycle assessments can improve the valley. So right here, this is the 17 Valley. You can see it's orange and red, which means that we need resources to help us be on the green. So here it is.
This is just a program by MIT, even though we're not sponsored by them. But this somewhere that you can check out. It's an 8-week course, and you can be a life cycle assessment expert and see the paycheck. So you gotta do something about that. So we wanted to take a moment and thank Beam Circular for giving us this opportunity. Alana Peach Fine for giving us this opportunity again, and also our mentor, Noah Hughes, for giving us his time. And so thank you so much. And don't forget to be awesome.
Any questions?